have the privilege to introduce Dr. Gustavo Dayan, Medical Director of Clinical Development at Sanofi Pasteur. He comes to his position with a great deal of experience. He received his MD at the University of Argentina, where he completed his pediatric residency and specialized in pediatric infectious disease. He served as CDC and Epidemiology Intelligence Officer, working as medical epidemiologist in the field of vaccines and vaccine-preventable diseases. He has several publications on polio, varicella, measles, mumps, and rubella. And that's a good thing for today. And served as a measles subject matter expert and MMR team leader at CDC. At Sanofi Pasteur, he provides strategic and scientific direction of the clinical development of vaccines, concentrating on West Nile and dengue. So today he will tell us why we need to stay informed about dengue and what's being done in the vaccine development. Dr. Dayan, welcome.
All right, well, thank you very much. It's really a pleasure for me to be here, and thank you for the invitation. So in the next few minutes, I'll try to summarize a little bit what is dengue, the disease, and the vaccines that are in development. So what we will do, we'll go through the virus, the disease, the public health burden of dengue right now, and then the vaccines, the different kinds of vaccines that are under development. And then at the end, I'd like to focus on the Sanofi Pasteur dengue vaccine. It's not that's because I work there, but it's just because it's the most advanced in the development. So to start, dengue is a flavivirus, so it belongs to the flavivirus family, as well as some others like yellow fever, Japanese encephalitis, tick-borne encephalitis, and West Nile. And this is a single-stranded RNA virus. And if you look at the structure there, it has 3 structural or genes that code for structural proteins and 7 non-structural proteins. And the structural proteins are the most important ones because they're related to the immune response. There are 4 serotypes. There are 4 dengue serotypes, 1 through 4. And it is believed that the infection to one of the serotypes provides long-lived immunity to that particular serotype, but only a short-term cross-protection against the other serotypes. Okay. Something wrong with this? There you are. Okay. So what is this disease? So what is dengue? So most of the clinical cases, as you can see, are asymptomatic. So basically, dengue is described, the clinical manifestations are described as a pyramid. So most cases asymptomatic. In some cases, we have this non-specific febrile illness, and then we have what we call dengue fever. So dengue fever is a high fever for 5 to 7 days, usually accompanied by headache, myalgia, joint pain. Remember that this is called the breakbone disease as well. And then this is usually followed by 2 to 3 weeks of debilitating fatigue, which is especially important in adults. Then the next step is the dengue hemorrhagic fever. So most patients would recover after dengue fever, but some of them would develop the dengue fever. And this is a disease where the vascular permeability is increased. And then we can have some signs of for example, plasma leakage, and this can give ascites, this can give pleural effusion, hemoconcentration. Also, it is normally accompanied by thrombocytopenia, which can be moderate to severe. And in some cases, with some other manifestations like myocarditis, encephalopathy, or also hepatic manifestations. Case fatality rate without any treatment is 20%, and this is typically done in children. Then we have the dengue shock syndrome, which is the most complicated form of the disease, and this is, of course, with signs of shock, with a short pulse rate. I put here some pictures so you know how they look, how these patients look like. You can see here a girl in Thailand with this dengue fever. Sometimes it can be also, we can see a rash. And in the bottom part, you can see a boy with some complicated, some severe forms of the disease. You can see the palpable liver and all the manifestations of shock. And I didn't mention this, but you may know dengue is a mosquito-borne disease. The main vector is the Aedes aegypti. And this mosquito is a really very urban mosquito, and it's close to the households. So this is a cycle, an urban cycle, and this is the most important one. The disease can be, or this virus can also infect monkeys, but this is kind of a forest cycle, and it's not as important as this one. So it is considered, dengue is considered now the arthropod-borne viral disease with the highest morbidity and mortality. And it is estimated that 2.5 billion people are at risk in over 100 countries. More than 50 million people are infected annually, and 2 million of those, mostly children, will develop these severe forms of the disease. I put here a map so you can have an idea of the areas in the world that are at risk. Not surprisingly, these are areas, subtropical or tropical areas. This comes from an article recently published in the New England Journal. And just to give you an idea of the magnitude of the disease, these are the number of cases that have been reported to the Pan American Health Organization for the region of the Americas. You can see from 1980 until 2010 an increasing trend in the number of reported cases with outbreaks that occur every 3 to 5 years. Interestingly, it's really increasing, and in 2010, the PAHO, the Pan American Health Organization, considered that there was really a pandemic in the region. So all the countries were really very heavily affected. Same thing, but just to show you the incidence, it's not only the number of cases that is increasing, but also the incidence, and you can see in the 80s, in the 90s, and the 2000s, and in 2010, which was that really big year. So, how about WHO? What does WHO think about this disease? So there is a WHO neglected tropical diseases group, and this group met in 2010 and established some targets and milestones for control of some neglected tropical diseases. And they set 2 goals, which is to reduce dengue death by 50% by 2020, and to reduce dengue morbidity by at least 25% by 2020. So how about prevention? So currently we don't have any prevention other than vector control and personal protection measures. But as you can imagine, these are really measures that are very difficult to maintain and really very expensive. So the development of a vaccine is viewed now as a public health priority. And just a little bit on the history, now just getting into the vaccine part. The history of dengue vaccines. So the first attempts were in 1928, in 1930. Then we have an important attempt in 1944 and 45, and this was a monovalent live attenuated vaccine developed by Sabin. Then after that monovalent, then we had the tetravalent vaccines that were also live attenuated, but they didn't work, and we'll see why they didn't work. I'll show you some aspects of this. And then they decided to embark on a different construction. So these are the chimeric vaccines. Chimeric vaccines basically combine different viruses to attenuate the wild-type virus. And they started with some monovalent vaccines and then the tetravalent ones. And why is taking that long to get a dengue vaccine? So what are the challenges? Well, to start with, we have 4 different serotypes. So to protect against disease, the vaccine has to have the 4 serotypes. And there are some, of course, technical difficulties related to that, because some of the serotypes may inhibit the growth of some others. So the replication may be increased or decreased because of these interactions. And of course, we need protection against the 4 of them. The other thing is that we don't have really an animal model. So Monkeys can be infected, but the viremia is much lower, and the disease is not exactly the same as in humans. So basically, we don't have that. There is another risk, which is what we call the risk of immunoenhancement after sequential infections. And what is this? So I don't know if you've heard that in some cases, the secondary infections may be more severe than the first infections. And this is related to an immunological mechanism. So basically, after the first injection, or the first infection in reality, the patients may have some antibody titers that are not neutralizing or partially neutralizing that could facilitate the entrance of the virus to the cells after a secondary infection. So this increased amount of virus in the cells would produce an increase in the cytokines and chemokines, and this would create an immunologic response that would create this increased vascular permeability. So this is it. This is a theoretical risk, but it is there as well. And then the other, and I would think the most important thing, is that we don't have a correlate of protection. So in some cases, some diseases or for some vaccines, we have a threshold we have to reach in order to be protective. But in this case, it is not known. And it could be even worse than that, because it may be different for the different serotypes. So after all this introduction, we'll see what different vaccines are being developed now. So basically, we have live virus vaccines. We'll see there are basically two different kinds. Inactivated vaccines, subunit vaccines, DNA vaccines, and vector vaccines. Let's start with the first group, which is the most important one, and these are the live virus vaccines. So basically, we have the traditional ones that are constructed through a traditional attenuation, and this is done through passages of the viruses in cell cultures. And we have different initiatives. One of them was the Walter Reed Institute, and then this initiative was followed by GSK. So in this case, it's traditional attenuation. What happened is that they started with a monovalent vaccine. This monovalent vaccine they developed a tetravalent vaccine. And this tetravalent vaccine was tested in Puerto Rico and in the US. But after some testing, after some phase 1 studies and even phase 2, the company, GSK, decided not to continue with this development because they had some technical difficulties in the production, and they wanted to have a vaccine with a different schedule, a schedule that could protect the vaccinees right away. So this is why it was discontinued. There was another attempt to produce a tetravalent vaccine, and this was originated at the Mahidol University in Thailand. Again, this is traditional attenuation, and at the time it was taken by Aventis Pasteur, which is a previous name of the company. And this was also tested in Thailand. Problem was that there were some safety concerns because one of the viruses, the dengue 3, was not sufficiently attenuated. Therefore, we had some reactions like a rash, fever, and this was considered not acceptable. Therefore, that project was totally discontinued. So that's the traditional attenuation. Then we have the recombinant vaccines. And there are several initiatives. So basically, one of them is the molecular attenuation through selective mutations. So what they do is they take the genome of the virus and they delete part of them, so 30 nucleotides. And this is considered a molecular attenuation. So they tested this vaccine, the monovalent formulations, and they detected that serotype 1 and 4 were really useful, safe, and immunogenic, but the other two were not. So what they did, they used these serotypes 4 particularly as a backbone where they inserted the genes of the other two serotypes, and they obtained this vaccine. This vaccine was originally constructed at the NIH and then taken by the University of Maryland, and currently is in phase 1. They have different formulations of a tetravalent vaccine, and it's also their plans to develop this vaccine in Brazil. Then there is another project or another initiative which is taking one of the dengue serotypes as a backbone and inserting the other genes coding for these membrane and envelope proteins. This was a vaccine that was created by the CDC, and then this project was taken by Immunogen, which is a developer right now. This vaccine is in phase 2 with studies in Colombia, and the phase 2 are in Colombia, in Puerto Rico, in Thailand, and in Singapore, with good results. Then we have the other recombinant vaccine, which is the one that uses the yellow fever vaccine virus as the backbone, where all these genes coding for structured proteins are inserted. And this project was originally developed by Acambis, and now it's being developed by Sanofi Pasteur. Then we have some other vaccines, like inactivated vaccines. So we all know how this works. So the viruses are cultured, and then they are totally inactivated. We have the one monovalent vaccine that was tested, and now there's a tetravalent vaccine that is going to be tested soon in a phase 1 study using GSK's battery of adjuvants, because these vaccines would require adjuvants. Then we have the subunit vaccines, and sorry if I'm being too boring. These are we have envelope vaccines, which they just take the immunogenic part of the virus, which is the envelope, and then they add the NS1. So these are the antigens that are supposed to produce immunity. Hawaii Biotech and now Merck has purchased that company, so they are developing this vaccine, and they have a tetravalent vaccine in phase 1. And then they have we have some others using the dengue E structural subunits being developed by the Genetic and Biotechnology Center in Cuba. This is also a vaccine that is a subunit vaccine, but it's still in the animal phase, so we don't have clinical studies for this one. And finally, we have DNA and vector vaccines. So these two have been originally developed by the Naval Medical Research Center, and they continued with the Walter Reed Institute. These are monovalent vaccines for now, and they are in phase 1 for the DNA vaccine. So, just in summary, we have in this table the vaccines that are currently, or the most important vaccines that are currently in clinical development, so that have been tested in humans. Phase 3, we have the Sanofi Pasteur vaccine. Then phase 2, Immunogen vaccine. The NIH is a tetravalent vaccine in a phase 1. As you can see, the most developed ones are the ones that are these recombinant vaccines that combine different viruses. And then this is an interesting, I wanted to show you. This is information coming from clinicaltrials.gov. As you know, every institution that is conducting a clinical trial has to publish the results in clinicaltrials.gov. This is a website that comprises all these clinical trials. So this information, you have kind of a registry of the studies or the clinical trials that are being conducted. We have phase 1 studies. As you can see, most of these are in green, are these monovalent formulations from the NIH that they tested. Really, they did a good job really testing different combinations and different possibilities for attenuation. Then we have phase 2. There are not that many. You have mainly Sanofi Pasteur and GSK with this old vaccine that I mentioned, and now Immunogen. And then you have phase 3, which is Sanofi Pasteur, which is the only one that is in that level. So let me explain you a little bit more in detail how this vaccine was constructed. So basically, they took both viruses. So in green, the yellow fever vaccine virus, and in blue, the dengue virus. So what happens is that they take the yellow fever vaccine virus as the backbone, and they take out basically the genes that are coding for the structured proteins, and they replace them for those of dengue. So the result is a combined virus that has these two different genes from yellow fever and dengue. So these combined virus are then cultured in viro cells, and we have then these combined virus which has the reproductive machinery of yellow fever, the yellow fever vaccine virus, but expresses the proteins, the membrane and envelope proteins of dengue. And this is done for the 4 serotypes. So for dengue serotype 1, 2, 3, and 4, and then we get the full vaccine. So what are the vaccine characteristics? So this is a vaccine that, as you know, it's a live virus vaccine. It has a stabilizer, but it doesn't have any preservative or antibiotics or adjuvants. And it is injected subcutaneously with 3 doses, 0, 6, and 12 months. And the indication is really to protect adults and children living in mostly in endemic areas. People living potentially in Asia Pacific, Latin America, and the Caribbean, which are, as you've seen in the map, the most affected areas. And this is just a few words on the clinical development. So the countries where Sanofi Pasteur is developing clinical trials, there are some phase 1 studies, USA, Mexico, Philippines, phase 2 studies in all these places, one ongoing proof of concept efficacy in Thailand, and one proof of concept with co-administration in Asia as well. And then we have phase 3 studies, lot-to-lot consistency, safety and immunogenicity. And also we have 2 ongoing efficacy trials, one in Asia and one in Latin America. Just to give you an idea where we are with the safety database, we have over 28,000 subjects that have been immunized with this vaccine. We really haven't seen any problem with the safety of the vaccine. The reactogenicity profile is very similar to the one of the control vaccines. We don't see any increase of reactogenicity in flavivirus immune subjects after the 2nd or 3rd doses, or in younger subjects. We haven't seen any dengue-like disease like we did with the other live attenuated vaccines. And really low vaccine viremia, which is also something interesting, and similar rates of adverse events and serious adverse events compared to the control. And this is immunogenicity. For immunogenicity, you see these are the subjects for whom we have immunogenicity data, around 2,800. And these are some of the results of the phase 2 studies that were conducted. So we have the first study in the USA, then in Vietnam, Peru, Singapore. We tried to do some studies in different endemic areas in both regions. And you can see the seropositivity rates and the GMTs on top. This is good, but we still don't know how protective this is, because we know there is some immune response, but we don't know how useful this is to protect against the infection. So in summary, for immunogenicity, we have a balanced immune response. We see a stepwise increase of seropositivity rates after each of the doses. Higher immune response in children, that's something that we have observed as well. Also a higher response in those that had received some flavivirus vaccination like Japanese encephalitis or yellow fever. And also higher titers in those that had been infected before with dengue. Just to give you an idea, so we have the first efficacy trial, as I said, in Thailand with 4,000 subjects, in subjects aged 4 to 11 years. And then 2 efficacy trials, one in Latin America and one in Asia, that are ongoing, with 20,000 one and 10,000 subjects the other one. You have the countries there. And just to finalize, just the conclusions, you see that dengue, as I show, is a disease with an increasing burden. Several vaccines are in development to protect against this disease. Sanofi Pasteur is the one most advanced. And then we have, well, this vaccine has approximately 20 clinical trials with more than 20,000 subjects, with a good favorable safety profile and an immune response induced against the 4 serotypes. We have a first efficacy trial in Thailand. We'll have the results soon, later this year, and 2 major efficacy trials, one in Latin America and one in Asia. And just to finish, we believe that the morbidity and mortality reduction of this disease is based on different factors. One is the adequate diagnosis and treatment of the disease. For sure, we have to recognize the disease and treat it. Then the mosquito control, that really plays a role, and the future vaccines may play a role in the future if they are developed. And this is the end. Thank you very much.
Dr. Dayan, we have two questions. What major causes or influences are they causing the increases in dengue attributed to?
Okay, so lately it has been an increase in the number of cases, as I showed, and this is globally. And the most important cause is really this increased growth of the urban populations. And in these areas, it's not planned. So we have big cities, and they are increasing the size. So this is one of the most important factors. The other one is the non-control of the vectors. So the mosquito population is really increasing. And we've seen in the Americas there was an eradication at some point, but it fell through, really it fell apart because it was very difficult to maintain. It's very expensive, so it was very difficult to maintain. Another factor is traveling. You know, traveling is increasing every day. So these traveling also transports the mosquito and of course transports the virus. And also some authors say that the global warming may also play a role. So these are some of the factors that really increase dengue incidence.
The second is, while the U.S. has some cases, are there any projections for increased incidence in this country?
Okay. Well, that's a tricky question. You know, in the U.S., there was, believe it or not, we did a review now with the PAHO, with the Pan American Health Organization, on the history of outbreaks of dengue in the Americas. And we, believe it or not, in the U.S. there were dengue cases early from the 70s, the 1780, I think. Those were when the first cases were reported. And then after that, mostly they were reported in port areas. So in the Gulf of Mexico, and also in the States, in Georgia, and even in North Carolina. So these outbreaks were related at the time with the trade. And at the beginning it was the slave trade, and then it continued to expand. Lately, we have cases in the Texas and Mexico border. These, of course, the mosquito doesn't respect the borders. And there was another one, a big outbreak, well, not a big outbreak, but a special outbreak in the Key West in 2009, 2010. And there were some cases in people who lived only there and have never traveled, and they never moved from the area. So I think there is an increasing trend for all the factors that we've discussed, and particularly traveling in the case of this country is an important one. So I think there will be also an increasing trend. I don't know if this has been quantified, but we can expect that. Any question?
Okay, we have time for one. Do you have the microphone? Okay, we'll just have to repeat the question. Okay, go ahead.
Yeah, this is, of course, the question was, we have in the vaccine, we have a schedule with 3 doses, and these 3 doses are separated by 6 months. So for travelers, this is really very inconvenient because they need to know in advance they will be traveling and to be vaccinated and to be protected. And yes, this is a problem we have with this vaccine because the schedule is really very long and it has 3 doses. So there are different studies in the plans to try different shortened schedules. And we know that in some cases, particularly for travelers, if they are vaccinated with some other vaccines like Japanese encephalitis or yellow fever, the immune response really goes up much quicker, and really they can be protected earlier than after the finalization of the schedule. So there are different ways to try to shorten the schedule. And for sure, we'll have some more data when we see the efficacy data and see if what the protection is after 1, 2, or 3 doses.
Thank you very much. We now have a website to watch and monitor the trials, and we'll stay informed about dengue. Thank you very much.